This chapter is going to focus on drugs used to treat multiple types of infections. Hundreds of years ago, young children would die frequently of what are now treatable infections. The earliest of drugs came in the 1940s and drastically improved the lifespan of individuals. Although drugs have decreased the occurrence of some infections, they haven't wiped out all diseases and probably never will. We may in fact be close to a post-antibiotic era where the drugs we have aren't working anymore. The goal of antimicrobial chemotherapy is for the drug to target the microbe without hurting the host. The drug needs to be easy to administer, and it needs to be able to find the microbe. It needs to be toxic to the microbe and non-toxic to the host. The drug needs to remain stable in the body for a long time and must be broken down and removed. Much like a germicide needs to have desirable characteristics, so does an antimicrobial. The drug must be toxic to only the microbe and should be microbicidal in order to kill the microbe. The drug needs to be soluble, meaning it should work even in the presence of body fluids. It needs to be broken down. It needs to not be broken down too early or removed too early. The drug should not lead to resistance and should work alongside the immune system. It needs to stay active and easily make its way to the microbe. The drug should be affordable and not cause allergies. Here are some definitions of various antimicrobials that we will be discussing in detail. Antibiotics are naturally produced by both bacteria and fungi. They do so during antagonism to eliminate their competition. The majority of antibiotics come from the bacteria Streptomyces and Bacillus. The Penicillium and Cephalosporium molds also produce a significant amount of antibiotics. Before an antimicrobial treatment can begin, a physician must know the type of microbe causing the infection. If it's bacterial, it will require different treatments than a virus or a helminth. The susceptibility of the microbe should also be known. A physician needs to know what drugs are going to be effective. The health of the patient also needs to be known. It's important to identify the microbe immediately. This needs to happen before the drug is administered. Examination of substances from the patient may be used for detection. Oftentimes, physicians will make informed guesses as to what the microbe is. This is done so as not to delay treatment. Certain organisms like Staphylococcus, Neisseria, and Enterococcus need to have sensitivity tests performed immediately. Fungal and protozoal infections don't require susceptibility testing because the antimicrobial drugs generally target all members in these groups. One method used in antimicrobial sensitivity testing is the Kirby-Bauer method. The bacterium is spread all over the surface of a Mueller-Hinton agar. Discs that are saturated with antibiotics are placed on the plate. A zone of inhibition or clearing around the disc are measured, and the antibiogram provides information that is used in choosing the prescription. It does not work well for fastidious microbes or anaerobic ones. Using the measurements of the zone, sensitivity or resistance can be determined. For example, if your zone around chloramphenicol was 20 millimeters, it's greater than 18, so the bacterium would be sensitive, meaning the drug was effective. In penicillin G, if the zone was 10 millimeters, that's less than 20, which indicates resistance. The penicillin G did not work on the bacterium. This is an example of a lawn of bacteria on the agar along with the disc. You can see the zones, which is what would be measured. test can be used on slow-growing and fastidious organisms. The entire strip has gradients of drug in micrograms. The MIC, or smallest concentration of drug required, can be determined by the mark on the strip at the end of the zone. The tube dilution test can be used to determine the minimum inhibitory concentration, or MIC. A series of dilutions are used to determine what's the smallest concentration required to treat the infection. What is seen in the in vitro setting or laboratory setting is not always what we will see in vivo or in living cells. A drug may not work because it doesn't have the ability to diffuse in the body. In the lab, there may not have been resistant microbes, and it may not have taken into account a mixed infection. Also, the patient may not have taken the drug as prescribed. Let's be honest, we've probably all stopped our regimen of antibiotics once we started to feel better. The therapeutic index is the ratio of drug that is harmful to us compared to the lowest concentration that is effective. We want a drug that has a high index. The lower the index, or smaller the ratio, the more of a chance there is for a bad reaction. Before an antibiotic is prescribed, the doctor must gather information. Any underlying health conditions must be known, including allergies to drugs or liver and kidney disease. The age of the patient also requires specific measures. Other prescriptions must also be known. Each and every case is going to be different, and it won't be cut and dry to where it's immediately known what to prescribe, as you can see in these two cases. The goal of antimicrobial drugs is to target metabolic processes in all types of microbes. It also needs to target viral replication. It should target structures necessary for microbes and be selectively toxic. It should only harm the microbe and not the host. A drug that has great selective toxicity is penicillin, which targets the cell wall of a bacterium. We don't have one, so it won't affect our cells. Those drugs that target the cell membrane are pretty toxic to us, and the more closely related the microbe is to our cells, it's difficult to have selective toxicity. There are several targets of an antimicrobial drug that can be deadly to the microbe. These include the cell wall, DNA, RNA, proteins, the cell membrane, and folic acid synthesis. This image details these targets as well as the types of drugs. We will be discussing each of them. Those groups of drugs that target the cell wall include penicillins, cephalosporins, and carbapenems. They also include isoniazid, which targets the cell wall of Mycobacterium. Don't worry about memorizing every drug, just focus on the three major classes. There are several ways that proteins can be targeted. Aminoglycosides prevent the mRNA from being read correctly, and an abnormal protein is made. Tetracyclines prevent tRNA from attaching to the A site during translation. Glycyclazines are derivatives of tetracyclines. Macrolides prevent elongation in translation. There are additional drugs like clindamycin that also target protein synthesis. Folic acid synthesis is also another target. Sulfonamides prevent the synthesis of tetrahydrofolate, which is necessary for not only folic acid synthesis, but also DNA, RNA, and amino acid synthesis. As I just mentioned, the sulfonamides directly prevent the synthesis of DNA and RNA. Polymyxins target the cell membrane by affecting phospholipids and causing proteins and nitrogen bases to leak out. There are classifications of antimicrobial drugs based on their spectrum. Broad-spectrum drugs target more than one type of group of bacteria. Narrow-spectrum drugs only target one specific group. Let's look at a narrow-spectrum drug. Isoniazid only works against mycobacteria and not any other groups. Carbapenems would be an example of a broad-spectrum drug because it works against gram-negative and gram-positive bacteria. The same goes for tetracyclines. We know that biofilms are difficult to treat. Typically, antibiotics cannot break through the outer matrix. So what do we do then if drugs don't work that well against biofilms? One strategy is to target the way that bacteria communicate. Daptomycin, which is used in deep tissue infections, is also promising. Researchers have also seen the addition of DNase with the antibiotic can help break down the extracellular matrix. Another method is placing antibiotics on devices that are being implanted in patients. We've talked about ways to target bacteria, so let's now focus on fungal infection. Those drugs against bacteria will not work against fungi. The problem with fungal cells is that they are very similar to our cells, so the antifungal drugs can be toxic to us. For this reason, there are only a few antifungal drugs compared to antibiotics. Macrolide polyenes target fungal cell membranes and can be used on yeast infections. Azoles target sterol synthesis and work against various types of yeast infections and AIDS-related fungal infections. Echinocandins target cell wall synthesis and can be used to treat Candida infections. Allylamines inhibit synthesis of enzymes necessary for ergosterol. This drug can be used to treat ringworm infections. Let's look at antimalarial drugs which target protozoans. Quinine used to be the go-to treatment, but less toxic drugs like chloroquine have been used lately. There are many stages in the life cycle of Plasmodium, so it's a bit difficult for just one drug to work. Artemisinin is also used to treat malaria. Let's look at other antiprotozoal drugs. Metronidazole is used to treat Entamoeba histolytica, which causes gastrointestinal issues. It also works against Giardia and Trichomonas. Helminths are a bit more difficult to treat because they're pretty large and because they're eukaryotes. The best drugs should target metabolism in all stages of their life cycle. Mebendazole and albendazole prevents the helminths from using glucose. Parentel paralyzes the muscles of roundworms and causes them to lose their grip on the intestinal wall. Praziquantel is used for tapeworm and fluke infections. Ivermectin can be used on pets as well as humans. Viruses are a bit different. They rely on the host cell, and if we target viral metabolism, we also target metabolism of the host. A lot of viral infections are prevented with vaccinations. The high prevalence of the flu and the common cold show the need for the development of more antivirals. There are three major modes of actions for antivirals. They include inhibiting the virus from entering, preventing synthesis of nucleic acids, and preventing the assembly and release. Let's look at a big problem we are seeing now. A lot of microbes are acquiring drug resistance. This can be something that occurs intrinsically, like spontaneous mutations, or acquired through processes like transformation. Resistance has been seen since the development of the first antibiotic. It was in the 80s and 90s that researchers started to see a significant amount of resistance. The resistance may come from those spontaneous mutations or via horizontal gene transfer. Chromosomal drug resistance often comes from spontaneous mutations, and sometimes a larger dose of the drug may be effective. Persisters exist in a population and go into a hibernation so that the antibiotic can't target them. Conjugation, transformation, and transduction all can transfer the R factor, which we discussed previously. The plasmids with resistance genes may be transferred, and transposons can also insert resistance genes. This then spreads like wildfire in the population. There are several mechanisms in which the organism can utilize these resistance genes. They can create enzymes that can inactivate the antibiotic, making it useless. They can decrease the rate at which the drug binds to receptors, thus decreasing how much drug comes in. Some bacteria can insert pumps into their cell membrane that can immediately spit the drug out upon entry. A bacterium can alter the binding site of the drug and prevent it from exerting its effect. And last but not least, if a drug alters a biochemical pathway, the bacterium can find another route to get to the desired product. Within a population, only a few cells have drug resistance. If the drug is not present, those resistance genes won't be expressed. Once the resistance organisms are exposed to the drug, the genes will be expressed and passed to their offspring. The resistant form will then dominate the population. We can see the small resistant population, and once an antibiotic is administered, the non-resistant forms are killed and the resistant ones survive. The resistant forms multiply and can transfer those resistance genes. In 2013, the CDC detailed a severe antibiotic resistance situation. There may be a point in time where all the drugs we have may be ineffective. Development of antibiotics is not something being done because it's extremely expensive and doesn't make pharmaceutical companies as much money. It also takes years to develop. Urgent threats include C. diff as well as drug-resistant Neisseria. The serious threats include resistant Campylobacter and Candida. And finally, the concerning threats are vancomycin staph aureus as well as resistant Streptococcus. If new antibiotics are not being developed, what is being done? Pieces of RNA that control expression of genes can be used to shut down metabolism. It's currently being researched in viral infections like hepatitis C and RSV. The insertion of defense peptides in the cell membrane is also being studied. CRISPR, which is a defense system in bacteria to protect themselves from bacteriophages, is also being researched to treat antibiotic-resistant infections alongside the antibiotic. Researchers are working to grow up non-cultivable bacteria which previously have not grown in the laboratory setting. The hope is to grow them and harvest their naturally produced antibiotics. Probiotics, which are live organisms, can help the intestinal flora and can replace it after taking antibiotics. These include yogurts and is safe, and most of the time it works. They're also believed to be useful in managing food allergies. Prebiotics enhance the growth of the good guys in the intestine. Fructans help to prevent the growth of pathogens. Fecal transplant can be used to treat C. diff infections that are recurrent. Through the use of a colonoscopy, feces from a healthy patient are transferred to a recipient with hopes of recolonizing their intestine with good bacteria to compete with C. diff. Of course, we want the drug to be selectively toxic, but some people will still experience adverse side effects. Virtually every part of the body can be adversely affected by drugs. Allergies can occur after taking a drug. The drug stimulates the response from our immune system. Penicillin is the most common drug allergy. The first time the drug is taken, sensitization occurs and no reaction is seen. The second time it's taken leads to the allergic response. Our biota, or normal flora, helps to protect us against pathogens. However, we do have a few pathogens. Broad-spectrum drugs can wipe out our normal biota along with the pathogen. A superinfection occurs whenever our normal flora is decreased or destroyed. Let's say a woman has a UTI caused by E. coli. The physician will prescribe an antibiotic which kills off the lactobacilli in the vagina as well. This decrease in normal flora then allows Candida albicans to overgrow and cause a yeast infection. Other antibiotics can wipe out the normal flora in the colon, which leads to a C. diff infection. The white circles in this image are the normal flora, and the red circles represent C. diff. Patient takes the drug, and it wipes out all the good guys, giving C. diff the opportunity to thrive. This is why probiotics are important to take with antibiotics. In the past, antibiotics used to be prescribed at such a high rate, from anything to a cold or acne. Broad-spectrum drugs also led to resistance, but luckily physicians are aware of this and have changed their approach. A lot of countries don't regulate antimicrobial drugs. For instance, one could go to Mexico and easily pick up penicillin. The person feels a sniffle and starts self-medicating. This is a major factor that contributes to resistance. It's important for you all going into the allied health fields to understand that antimicrobials are great, but also understand their limitations.